High elevation conifers are vitally important to ecosystem health and function. However, they face grave threats in the context of a changing world. Our research group is studying the physiology of these organisms as a means to inform conservation strategies through improved scientific understanding. Our research is focused on whitebark pine, limber pine, and Great Basin bristlecone pine, which are all 5-needle pine species found in high-elevation ecosystems of western North America. These 3 species are all considered keystone and foundational to some extent. These species play a crucial role in creating and maintaining habitats that support other species. This is because they are able to establish in exposed, cold, windy sites where no other species would be able to grow. This establishment then provides protection from these conditions for other plant species to establish. They also provide habitat and forage for wildlife. Their large, nutritious seeds are a food source for a range of species, from grizzly bears to birds such as the Clark's nutcracker. On top of these benefits, which promote biodiversity, these species also provide valuable ecosystem services, such as the stabilization of snowpack, delay of snowmelt, and erosion control. These effects perform important regulation of downstream water supplies. However, their survival is currently in jeopardy. Indeed, whitebark pines have recently been listed as threatened by the U.S. Fish and Wildlife Service, making it the most widespread tree to be listed under the Endangered Species Act. One important factor leading to this decline is white pine blister rust, an invasive fungal pathogen native to Asia. Another key factor leading to this decline is climate change. The coming century is predicted to be marked by increases in global temperatures and more frequent intense drought events. In addition to the hydraulic stress this will cause, evidence suggests that climate change may exacerbate the effects of white pine blister rust. Together, these factors have already caused increasing frequency of mortality events, and these trends are predicted to increase into the future. Now, if you're like me, you may be asking yourself, what can we do about all this doom and gloom? The answer: there are 2 key conservation strategies for mitigating these negative impacts, which are assessing vulnerability to stressors and restoration through the outplanting of seedlings. However, to achieve these goals, we need to know more about the species' physiology. Physiology, or the way in which a living organism functions, acts as the interface between an organism and its environment. The physiological responses of organisms to changes in their environment will determine the nature of functions such as metabolism, growth, and reproduction. These functions are key elements in determining the persistence of a species in certain environments, which in turn determines the biodiversity of an ecosystem. Let's take a look at how that works. This box will represent an ambient environment. Within that environment, we will have 3 organisms with unique physiological traits. The first organism has physiological traits that confer abiotic stress resistance. For example, a tree exhibiting stomatal regulation that allows for high degrees of drought tolerance. We'll call this organism A. The second organism has physiological traits that confer biotic stress resistance. For example, a tree that creates metabolites which defend it from pathogens. We'll call this organism B. Finally, we have an organism with physiological traits conferring both biotic and abiotic stress resistance. We'll call this organism AB. In an ambient environment, all 3 of these organisms are happy, so they can reproduce and persist in the environment, promoting biodiversity. Now, let's see what happens in an environment that has vectors of biotic stress such as white pine blister rust present, represented by this yellow box. In this environment, organism A, which does not have biotic stress resistance, will likely experience mortality, while organism B and organism AB persist. Next, let's see what happens in an environment where both biotic and abiotic stress are present. This red box represents our predicted future, where these species will face both attack from pathogens as well as climate change-induced stress. In this environment, both organism A and organism B will likely experience mortality, while only organism AB, with its resistance to both types of stressors, will persist. It is therefore evident that for a species to be successful in our changing world, it will need both biotic and abiotic stress resistance. Much research has focused on biotic resistance in whitebark pines in order to identify seedlings with rust resistance for outplanting restoration efforts. However, there are still important knowledge gaps regarding the physiological traits associated with resistance to abiotic stress, such as drought, heat, and cold. This is important both for informing outplanting strategies and predicting what populations may be most vulnerable to future stress. This is where our research comes in. We'll be conducting a greenhouse-based experiment using unique seedling populations. We have 17 populations of 2, 3, and 5-year-old seedlings across 3 species, the parent material for which came from contrasting climates, ranging from hot and dry to cool and wet. Using those populations, we will impose abiotic stress, such as heat, drought, and extreme cold. We will then measure physiological traits in response to these stressors, including rates of photosynthesis, water use efficiency, growth rates, and carbon allocation. To understand why these are important, let's take a closer look at what carbon allocation patterns can tell us. In normal conditions, trees use energy from the sun to fix carbon dioxide from the atmosphere into organic carbon. This carbon can then be put towards a variety of functions, such as leaf and root growth, basal metabolic functions, reproduction, abiotic stress resistance, such as adjustments allowing continued hydraulic function during severe droughts, biotic stress resistance, such as production of chemicals that protect plants from attack by pathogens, and storage reservoirs. In non-stressful conditions, plants can undergo photosynthesis at rates that allow them to allocate carbon to functions as needed. However, in stressful conditions, such as periods of heat and drought, plants may be forced to reduce rates of photosynthesis, creating carbon-limited conditions. In these conditions, plants face trade-offs in terms of where their limited resources will be allocated. Carbon allocation strategies in these conditions can tell us much about a plant's resistance to stress, and therefore the likelihood of its future persistence. By measuring physiological responses, we can determine the relative abiotic stress resistance of our seedling populations. For example, Population 1 may be able to allocate significant resources to abiotic stress resistance in stressful conditions, so we'll shade it dark blue. Population N, on the other hand, may be less effective at maintaining abiotic stress resistance functions, so we'll shade it light blue. Using this data, we can then place our populations along a continuum from less resistant to more resistant. Using this knowledge of differences between populations in abiotic stress resistance, we will inform conservation strategies by providing insights into what populations may be most vulnerable to future stressors, and what populations may have the greatest success rates in outplanting restoration efforts. This will, in turn, increase the likelihood of species persistence in the face of future stress. Because these trees are keystone and foundational, conservation of these species will have cascading positive effects on ecosystem health and function.